Even the instruments of being a
rest.
physician, this is essentially an
It
instrument
would be
to test
good for
your
the
nerves,
digestion and
right?
absorption.
Because if you hold out your
So
arm, you
we
can
all
hold it straight
know
by
what that
having
means.
a constant
So I'm going
attention
to
in your
lead
receptors. This is that you're
meditation.
tuning for.
I think maybe for
But now
20
people
minutes,
like
because
Mary Lou
we are
Jepsen
running a little bit
can look
behind.
inside the brain through the skull with
I
infrared light.
heard
Like,
that
wow.
during the
a big
BrainMind
deal.
Summit at MIT,
So,
in
unlike
May,
Ernest
Dr.
Solvay,
John
you
Kabat-Zinn
can't solve
led
this
a
problem
mindfulness
and even
meditation.
organize it
Any of you
unless you organize the scientists
were
together,
there?
help technology transfer,
So when I was asked to lead
and filter a
practice, I
surplus
was
of
a little
ideas
bit
out there. It's
nervous
not that
and
there aren't a lot of good
hesitant.
ideas, it's
So,
like how do you
wow.
tell the ideas from each other?
Because I've been working with John
So
since
a
2011
flywheel is a cool concept.
when he teaches in China. And I know really, really well how it feels when he enters a space and leads a practice. So I decided, and I told Diana that I'm going to lead a different practice, not a mindfulness practice, but a loving kindness meditation. I want to see how many of you have practiced that. Okay. How many of you have been on a retreat, like 3-day, 5-day, 7-day, 3 months loving kindness retreat? Because people do that, actually. Okay, so said it's a different meditation, but there's still ongoing debate whether a loving kindness meditation is a type of concentration meditation through which we repeat short phrases of good wishes that are intended to people or intended to different categories of people, including ourselves, a benefactor, loved one, a neutral person, a difficult person, and to all beings. So through repeating these phrases, we start to gather our scattered mind and start to concentrate. So that's why some people think the loving kindness practice is a concentration meditation, but others argue otherwise. So a Buddhist scholar from the Oxford University, Dr. John Peacock, did a 2-day talk at the Insight Meditation Center in Redwood City several years ago. He talked extensively on loving kindness as one form of mindfulness meditation. So these short phrases being repeated really serve as pebbles dropped into the well, and the well of your very being. And when you're repeating these words, you pay very close attention to the experiences that's been evoked by these repeated phrases. So you not just listen to the words, you listen within the words and behind the words and beyond the words to really stay close to the experiences that these short phrases would bring to you. So, and loving kindness is translated from Pali word called metta. So the more accurate translation is a kind of boundless, expansive friendliness. So it's less demanding or sentimental than I needed to love everybody. Well, at least we can be friendly or befriend other people, including ourselves. So with that, I'm going to lead a loving kindness meditation. So I would invite you all to let go of things that you are working on. So letting go is part of the practice. Okay, so then we'll adopt an erect yet comfortable sitting posture. So I'll be sitting here as well. And if you feel comfortable, you can gently close your eyes or just soften and lower your gaze. So feeling your feet flat on the floor, your hands in the laps. You can also imagine as if there were a golden thread that's gently pulling from the top crown of your head. So your head, neck, and spine are properly aligned. So this sitting posture really embraces a sense of dignity and alertness. Now take a few full breaths. Breathe in deeply through the nose and breathe out through the mouth. Breathe in again and breathe out, and then follow the natural rhythm of your own breath. With a gentle attention, you can scan your body, letting whatever tensions that might be locked in the body somewhere. Loosen your shoulders, soften your hands, relax your belly, and ground your feet. And bring a very slight smile to the corners of the mouth. Even feeling a smile on the inside of the mouth. And expanding the smile to the corners of the eyes. Softening the flesh around the eyes. Expanding the smile downwards into your heart. Creating a receptive and tender space in the heart and chest area. Now I'm going to offer a few phrases, and I will take a brief pause between each phrase so you will have time to repeat the phrase either silently on your mind or can whisper. We'll start offering loving kindness to ourselves. May I be safe and protected. May I be well in mind and body. May I be at ease and happy. May I be filled with loving kindness. So as you repeat these phrases, be aware of feelings or images that might be arising this moment. And make this as an experiment. You can also find the words or phrases that speak the most to you in this moment. May I be safe and protected. May I be well in mind and body. May I be at ease and happy. And may I be filled with loving kindness. And paying attention to the echoes or the ripples once you dropped these pebbles into the well of your being. Now bring a benefactor to your heart. Perhaps you have many in your life. Find one, the one that you feel really cared for you, provide you opportunity, protected you, or invested you with love and other resources. And bring this benefactor's image to your mind's eyes and offering loving kindness to this benefactor. May you be safe and protected. May you be well in mind and body. May you be at ease and happy. And may you be filled with loving kindness. Perhaps you can imagine upon receiving these good wishes, the benefactor smiled and was filled with joy. And you can replace you with this benefactor's name as you repeat. May you be safe and protected. May you be well in mind and body. May you be at ease and happy and strong. And may you be filled with loving kindness. And now bring to your heart a loved one among your family members or your dear friends. Think about the qualities that in this loved one that you most appreciate. Recall this dear one's good energy, generosity, good humor or wisdom, and sending good wishes to this loved one. And again, you can replace the name, or you can create your own phrases. May you be safe and protected. May you be well in mind and body. May you be at ease and happy. And may you be filled with loving kindness. Of course, if you have more time on your own, you can repeat these phrases over and over and over again. and let the feelings just permeate your body and mind. So may you be safe and protected. May you be well in mind and body. May you be at ease and happy, and may you be filled with loving kindness. And now we'll be expanding the circle of loving kindness to include all beings, near and far, humans, animals, trees, flowers, the earth, the universe. You can imagine as if you were a beacon or a lighthouse, grounded in the midst of waves and even storms on the ocean, and radiating beams of loving kindness, this boundless friendliness in all directions. May all beings be safe and protected. May all beings be well. May all beings be at ease and happy. May all beings be filled with loving kindness. As you repeat these phrases, also knowing that you are not the only lighthouse, and there are many others surrounding you, radiating loving kindness to you as well. So if you might be willing and feeling comfortable, you can open your eyes and maybe stand up, just like a lighthouse. And you don't have to, but only if you feel comfortable. And then gently close your eyes again and really immerse yourself into this beaming, boundless, and expansive friendliness that you're giving, but at the same time you're receiving. So resting in the feelings that's been generated from this practice. And whether you are sitting or standing, again, bring your attention back to the body. Feeling grounded and sensing this body sitting or standing as a whole, breathing. So a breathing, beaming lighthouse. So I'm going to ring the bells. So just follow the sound. Check into your body and mind. And if your eyes are still closed, you now can gently open your eyes, and maybe if you're not shy, look around and greet the other lighthouses around you. And thank you for practicing together. Fernando. Yes.
One of the many things that we study at in my lab at Karma is a project on what makes a pleasant voice. And that to me was the single most pleasant voice I've heard in a long time. Absolutely beautiful. So tomorrow we'll go back to instrumental music when I present some of my music, but today's next musical interlude will be from my friend and colleague Fernando Lopez Lascano, who teaches with me at Karma. Fernando's office has an LP cover of Switched On Bach, which I think for him and myself and a number of other people of my generation that we work with was sort of the life-changing moment. Fernando embodies that in incorporating ancient technology with new technology. And the one thing that Nando has brought and has completely changed my life is an awareness and a control of space. So I think what you're going to hear now is as much about where things come from as what they are. Fernando?
Thank you, Fernando. So we're going to please another hand of applause for Fernando. So we're going to take a couple minutes here to let everyone back in from lunch, so just bear with us. Thanks. All right. Welcome back to the Blown Mind Summit. So, never doing that one again. So first, a little history for everyone. My name is Calvin Nguyen, and this is, as you know, Diana Seville. We are co-founders of BrainMind with Michael. And the first time this group came together was about 12 months ago. And we were amazed because the energy that everyone brought was inspirational, and also because we got started 2 weeks before the event. And so we had absolutely no idea what we were doing, but we knew one thing, and it was proven out, which is that there's a lot of latent energy in this sector from many people who care about doing good things. And it was only a need for an organizing energy to unlock that latent energy. And so with the cost of experimentation and research and building companies rapidly decreasing and becoming increasingly accessible, we just needed to combine and apply some simple adjustments to the system. And as you heard earlier today from Amit and from Michael, those things are being done today in this room. And the metaphor I love to use for this is adding a little bit of tin to copper, because the result is so much stronger that it seems to be composed of an entirely different metal. And so over the last 12 months, we've been inspired by the response of the community. We've introduced 155 different philanthropists and foundations to 284 scientists across the 3 sessions in 12 months. We've brought $250 billion in investment assets to meet with early-stage neuro companies. And from those meetings, scientists like John Gabrielli, who presented at MIT, received funding from 4 different philanthropists to do research like a 16th-month study on somatic work for healing stress in children. And actually, some of those people are in this room today. So if you would like to stand up, and when I call out your name, please do. So William Spear, thank you so much for supporting 4 different people that he met at MIT, including a nanotechnology for that Anita Goyle is building for accurately detecting genetic fingerprints from any biological organism, a closed-loop neurofeedback device to help people sleep, a nonprofit to help match entrepreneurs with groundbreaking science. It's reported to us also that Jay Sanguinetti is also in the early stages of getting funded for his ultrasound research to actually help us reach advanced states of mind faster. But most exciting, I think, is actually the entrepreneurs who started working with scientists to build companies. And so Amal Sarva worked with Neural Shaw to actually turn his fundamental research on the sex difference in brains into a business that can help us better understand ourselves and our behavior. And Ben Lowenstein and Paul Litvak from Airbnb left their jobs after the last summit to build a company with Nick Malashier at Stanford, using household objects to collect biodata to help us better build drugs and better diagnose diseases. And you heard earlier today that Dr. Spiegel had met Ariel Polar 2 years ago. But what he didn't tell you was the night they met, Ariel Polar went home and stayed up all night to write the code for that Alexa app that he showed today. So these type of things that happen at these summits, this isn't something that is just a happenstance. I think it's because people come here, you come here, and we ask you very specifically to build these things, and we're really inspired and excited about the things that will come out this weekend as well. So I also want to share the stage with Diana to talk about the Asilomar and Neuroethics Initiative that we've been working on this last year.
Yeah, so Michael mentioned a little bit about this in his talk. I wanted to give a little bit more detail in case anyone here is interested in getting involved. It all started about 2 years ago when Ed Boyden was really inspired by the 1975 effort to protect humanity from the potential downsides of genetic recombination. He said, we should do that for the brain. Now would be a good time. And we knew as an organization that this would be important. If we're going to find new pathways to build high-impact companies in brain science, we better have a pretty good understanding collectively of what is a good idea and what are some ideas that we should be cautious about. And so I had a chance to speak about this with Wendell Wallach and Phil Rubin at the last BrainMind meeting at MIT. They pointed me to Jonathan Marino, who did a survey of the entire field of neuroethics and its history, surfacing the people that were most passionate and engaged in this space. And something that I realized throughout the conversations over the summer with people that are active in this space is it's very difficult to match their energy and passion and good intent. You know, our own Laura Roberts was one of the first to get involved, and she has her own expertise. She's been active in the Brain Initiative Neuroethics subgroup. Steve Hyman from the Broad Institute from Harvard has agreed to chair our steering committee. Nita Farahani, who's the president-elect of the International Neuroethics Society, is going to co-host our first planning meeting at Duke at the end of February. Carolyn Montojo, who's here today from the Kavli Foundation, is helping us to build a coalition of funding support to make this happen. And the group keeps getting stronger. Tom Insel, Kara Ramos, Martha Farah, Hank Greeley, Christine Grady from the NIH, and Karen Rommelfanger from the International Brain Initiative is going to help bring international stakeholders to the fore. So we have a major opportunity here to do something unprecedented, to do a decadal review of neuroethics for this space, not just for academics, but for investors, for the private sector, for entrepreneurs, for the stakeholders of the BrainMind community. So if you are interested in getting involved, I hope that you come and speak with me about it. And also you should join Laura Roberts' brainstorming table on the subject tomorrow afternoon. Thank you.
Thanks. All right, so moving on to the Entrepreneur Spotlight. We brought these 4 companies together today because they fit in that top part of the 2x2 square that Michael showed. These are highly impactful companies, some of which, and maybe all of which, have venture-like returns, but also have wide-ranging implications for the way we even do research and the way that we discover drugs and deliver them. So the first person we have today is Kenneth Irving. Irv is a co-founder of Rocket Science Health. Irv tells me that he is passionate about protecting human rights, especially with personal data use, promoting brain health, and preserving water habitats and biodiverse soils. But in his words, is the best way to hear about it, his favorite way of enjoying nature in the high Arctic is hiking, rowing, and canoe tripping. You'll have to ask him what canoe tripping is. He is best known for his 25 years leading an old world industrial concern, as he calls it, at the largest energy company in Canada, and founded this most recent company to address a 21st century problem. He's one of the most interesting people I've ever met. Please welcome to the stage, Irv Irving.
Thank you. We had some great meditation and some mind-blowing sound, and I feel I should break out into some modern dance now just to keep your attention. I grew up in a family business, and I admired, I loved, I was completely taken by my grandfather. So when the day came that I was asked to lead the company that he founded, it was a dream come true. And I worked very hard for a number of years, staying focused on the business, growing the business, and ignoring extraneous circumstances that ultimately was impacting my health. And one day I ended up in the hospital. And I was very lucky to have had excellent health care, including access to intranasal ketamine for my treatment-resistant depression. And as a patient, I really feel that it saved my life. So several weeks later, though, I was home. I was taking my prescribed number of sprays, and I overdosed. The medics who came to my home assured my wife, Tasha, that my out-of-body experience wasn't life-threatening. But I was intrigued to find out that over the course of the next number of weeks, when I was doing a bit of research, that the accurate dosing for CNS drugs is a problem. Now, while direct-to-brain medication is being promoted as the future for CNS drugs, it seems investments in biologics, in small molecules, in personalized medicine might be getting out ahead of devices that can deliver these drugs directly to brain. So, inspired by my grandfather's sense of entrepreneurialism and his ability to form partnerships, I decided to dig in, just to jump in and see what I could find. Now, to an oil guy, when you look at a nasal device nozzle, it looks a lot like a feed nozzle to a petrochemical plant. One's a little larger than the other. But the idea of feeding an accurate payload of hydrocarbon to a specific point high up in a steel vessel is not an uncommon problem. So I put some engineers, oil engineers, in a room with a medical device company, and from there we blended in a little bit of agile development protocols with medical industry protocols and regulation, and that's really how our story started. Now, it turns out that the lower nasal anatomy does a fantastic job at redirecting particles from reaching the upper part of our nasal anatomy way up high in the olfactory region where drugs have a direct pathway to our brain. And that's why spray devices quite often are very inaccurate at reaching the olfactory. If you're looking at this slide, the best way to interpret it is our technology is able to deliver 5 times more drugs to the olfactory at 1/5 the variability of the current state of art. It makes you wonder how many trials might have failed because of dosing accuracy versus the efficacy of the molecule itself. Earlier this year, we conducted in-human studies, and it was determined that our unconventionally derived technology could deliver a bolus of cohesive bolus accurately and repeatedly to the olfactory region. And we were pretty excited about that, and we continued to work on that technology, focusing more on human factors and keeping device costs low. Now, while we are a commercial enterprise, we have committed ourselves to 4 priorities that we wish to make an impact on. And we have gotten out at the very early stages of phase 0 to speak with researchers, government agencies, NGOs, to get advice on our social priorities. And that really did lead to a lot of innovation that has made our device a much more effective platform that can deal with a much broader spectrum of CNS disease states and use cases. We believe that our device can deliver and make accessible important CNS drugs for individuals needing them. We often get asked, for example, would our device and its accuracy using generic ketamine be as effective as a patented derivative molecule delivered by a spray device? And we think the answer is yes, and at a fraction of the cost. We're currently at the stages now where we're looking for non-dilutive funding or mission-driven funding with organizations that share our same social priorities. We do intend to license our technology to organizations that have molecules that could benefit from more accurate dosing, and we would welcome preliminary discussions in that regard. Now, as an outsider to neuroscience, I'm in awe of what we know about the human brain and what's left to discover, much like an astronaut might feel when looking back at the Earth when in outer space for the first time. I'm grateful to be on the stage in front of you, and I just want to say I'm particularly grateful to be in front of some of the people in this room that have had a direct impact on the fact that I'm standing in front of you here today, healthy. Thank you.
Thanks, Irv. So next is Matt Engel, the founder and CEO of Paradromics, and maybe one of the bravest entrepreneurs to walk up here today because his company that is building a brain-computer interface is presenting to a room that has Neuralink, kernel, and control labs all in it. So Matt has a long history of disruption, starting with his first business in grade school where he was so disruptive, he was disciplined by his teachers. He has a PhD from the University of Heidelberg, where he worked at the Max Planck Institute for Medical Research. And prior to Paradromics, founding Paradromics in 2015, he was a postdoctoral researcher at Stanford University in the lab of Nick Milosh in the Department of Material Science and Engineering. Matt Engel, the stage is yours.
So thank you for having me here today. Many of you have heard about brain-computer interfaces. Maybe some of you haven't. A brain-computer interface is an implantable modem that goes in your brain and allows the brain to connect to a computer. For those of you who have seen press reporting or have been paying attention, there's been a lot of buzz about what brain-computer interfaces may do in terms of human augmentation, even human transcendence. Some people are even interested in a war with artificial intelligence. All of that talk can give you the idea that brain-computer interfaces are kind of far away, maybe science fiction. I think that can't be further from the truth. There's a lot that BCI can do right now, and at Paradromics, our vision for brain-computer interfaces is much more near-term, but I think it's just as audacious as some of the other things that you might have read about. We see brain-computer interfaces as a way for brains and computers to directly exchange data, and by doing that, we can reframe a lot of currently untreatable physical and mental health problems as solvable data problems. So suddenly, once you can connect a brain to a computer, a whole ecosystem of computer-connected devices becomes your new neuroprosthetics industry. Brain-computer interfaces for healthcare use this isn't a niche application on the way to cyborgs. This is the big show. There are millions of Americans today who would benefit from advanced BCI. Just to take the top headlines: speech and motor prosthetics for people who are paralyzed, sensory prosthetics for people who are blind or deaf, and even using bidirectional BCI as a way of treating currently untreatable mental health. As a startup, we're specifically interested in helping one group of people that we think is still a significant market share from a company perspective. We want to build a data link that will allow for paralyzed patients that can't speak or type to communicate with the world. We think this is the right place to start because of the very high unmet clinical need, but also of the really powerful clinical proof points that we've seen in clinical trials over the last decade. And since we're at Stanford, I would be remiss if I didn't talk about the BrainGate program, and especially the work that Paul Nuyazhukian, Jamie Henderson, and Krishna Shenoy have been doing here in allowing patients to use what's called the Utah array, which is a probe that has 100 electrodes to plug into a device like a tablet and to start texting after they weren't able to communicate for several years. Now, these devices that are being used in clinical trials right now have their problems. They're not exactly productized. One of the problems is data rate. 100 electrodes isn't enough to constitute a profound brain-computer interface. Individual neurons that are recorded, roughly say 100 neurons to 100 electrodes, they have a data rate that's roughly equivalent to an old-timey telegraph operator. And so if you want to build a high data rate device, you have to record from lots of neurons. The state of the art is 100 electrodes. We see that we can build devices that have 100 times that number, so 10,000 electrodes per square centimeter. There's also a lot to be done in productizing the device so that instead of being only usable during a research setting, it can be taken home, it can be unobtrusive. There are 3 core things that we're doing over and on top of what the Utah array is doing right now. One of them is electrode density. So we're delivering 16 times more electrodes per unit area versus the state of the art. Another is that we're making it minimally invasive, both minimally invasive in the sense that each individual electrode is 4 times smaller than the equivalent electrode shank in the Utah array, also minimally invasive in the sense that everything is under the skin and it can be worn in a way that doesn't substantially impact the patient's life. The third point that I'd want to talk about that is relevant, I think, across the field right now as people look at the way that we should be interacting with brain-computer interfaces should be interacting with tissue is that we're building it out of robust materials. The 3 most common ways that interfaces fail right now are because of polymer insulation and because of metal thin films falling off. A lot of proposals to build flexible electronic arrays that would do the same thing that we're trying to do are based on these thin films and these polymers. We're looking to have a device in clinical trials in 2022, and you can see the work that we have cut out for us, but we think that there's a a very plausible path ahead for a company. I want to update you on 3 quick areas of technical progress that we've made in our kind of core competency areas. One of them is in hardware, the other is the in vivo recording that we've been doing, and finally on real-time data processing. Paradromics currently has the highest capacity neural recording system in the world. To talk a little bit about the front-end architecture, we're building arrays of up to 10,000 electrodes. We have a back-end that's capable of reading out 65,000 simultaneous channels. We've been deploying this in preclinical setting, meaning animal studies. Here on the right is an example of a recording from 791 neurons in a rat. And then just to tease it a little bit, over the last couple of weeks, we've started inserting arrays of thousands of wires into sheep. So that's an example of an activity map from 5,800 active microelectrodes. One of the most common questions that I get after I talk about the scale on which we're acquiring data is whether we can analyze data at that scale. And the answer is not only yes, that we can, but also that we can do it at acquisition speed. So I know I'm out of time, so I won't belabor this. One of the most common things that you do with a data set like this is that you detect all of the action potentials, which are the currency of information in the brain, and you extract salient information from that. We can do that for 10,000 channels in real time. Not only that, but our implantable device that will be complete at the end of the summer of 2020 includes analog electronics that allow you to do all of this data processing in the chip itself at a power bill that allows you to still implant it in the brain. So I'm happy to talk with anyone who has more technical questions for me or commercial questions for me after the talk. Thank you.
Our next group has been in stealth for a few years now. They were best known for raising $40 million a few years ago, and a website that has just 2 words on it. Sean Escola is co-founder and CEO of System 1 Biosciences, an assistant professor of psychiatry at Columbia University, trained in computer science, biochemistry, neuroscience, and medicine, all at Columbia. He's board certified as a psychiatrist and experienced treating patients with severe brain disease. And his research focuses on understanding the computational underpinnings of the emergent complexity in neural networks that underlie behavior, perceptual, cognitive, and emotional functions of life. So, for example, Sean had a pet rooster when he was a kid, and his parents took it away because it pecked at him too much. But Sean would still like another pet rooster someday. So can you explain that one, Sean? In System 1, Sean unifies his 2 professional pursuits to characterize his computational dimensions of the disease directly in human circuits. So please welcome to the stage, Sean Escola.
Hi, I want to thank the BrainMind Institute for having us here. We've been a company for 3 years. This is the first time we've made a public presentation. I'm really glad I had the opportunity to do it here. So at System 1, we're interrogating how we can build a phenotypic drug discovery platform. And why do we need to do this? Well, drug discovery is hard, as everyone here knows. Over the years, we haven't really increased the number of drugs we're producing every year. That's in red. But the amount of money we're spending per drug that we do produce is rising astronomically. And so we're having diminishing returns. Another way to think about these data is if you look at the probability that a drug that came out of a preclinical discovery program actually makes it through the clinic, it's extremely poor across all of medicine, but very poor in particular in the brain. And so what does this mean? Does this mean that we have found all the easy drugs and it's just really hard news from here? I mean, maybe that is the case. But actually, we think that there's a different answer. We think that we know that many drugs, especially in the brain, have been discovered serendipitously. They were tried for something else, or some intrepid psychological adventurer tried it on themselves, and we learned something about the brain, and we're able to develop drugs that have helped patients. And in fact, there are large libraries of neuroactive compounds. This is one of them. There are many others. You could order thousands of neuroactive compounds if you wanted. I would argue that the biggest problem that we have is how do we map these compounds and potentially combinations of these compounds to the complex neurological and psychiatric diseases that our patients experience. So why are things hard when it comes to treatment in the neurosciences? Well, of course, you have just so many different levels of complexity. One of them is the genetic complexity. There's no single gene for many of the disorders we're interested in that allows you to explain the disease. And similarly, we use very poorly matched models. The mainstay of discovery in preclinical research is the mouse, and there are features of primate neurodevelopment that are completely absent in the mouse. And finally, even if you had a handle on the genetics of a disease and you felt like you had a good model, neurobiology is just rich and complex. And let's walk through some of the complexity and try to get a handle into how hard this problem is. So of course, you can start with the genome. It's big, but it's tractable. The genome implies a gene expression network that occurs in all the cells in the body. And these cells, of course, they come together and ultimately arise the process of from that ultimately arises the process of neurodevelopment. And this is an unfolding process in time where you get a diversity of cell types and all of this rich structure and different layers and architecture. And the neurons in this architecture, they wire up to each other, they become a neural network, and action potentials start firing through a network like this, and they're processing information. They're sending information around in this system, and ultimately it's that capability of the brain to do information processing that gives rise to the features of human experience. And so I'm a psychiatrist. If a patient comes into my office and says, well, I've got a symptom, that symptom is always going to be in the emotional, perceptual, cognitive, or behavioral domains. And so the question that I have then as a scientist is how do I map back through all of this complexity in neurobiology to figure out why this patient has one of the symptoms that they're reporting? And I would argue that what we need to do is we need to have human models, patient-derived human models, that we can measure in many, many different ways and get rich data sets from, and ultimately characterize those data sets through the powers of, I will mention, of machine learning to get better descriptions, richer descriptions of disease than the ones that we've been able to characterize thus far. So if you want to use a human model today, you do this. You take neurons in a dish, or maybe not even neurons in a dish. And of course, if you compare this to the complexity that I just laid out, there's a huge mismatch. So this is the organoid. And this is a paradigm shift in our ability to do rich, complex disease modeling in a dish. Organoids have, they're derived from stem cells, which you can get from patients, and they have much of the neurodevelopmental biology. They have many of the cell types, and they have the rich, complex activity of intact brain. They're not intact brains. It's just a major step in that direction. So if you look on the left, you see some of those same developmental features that I pointed out before, and on the right, the rich, complex activity, and this is spontaneous activity in the organoid, but if you look at its structure, it's very similar to the kinds of things you'd see in spontaneous activity in vivo animal models. The problem, of course, is that organoids are very variable. They're variable across many dimensions. They're variable in their cytoarchitecture, they're variable in their gross morphology, and they're variable in their neural activity, in the neural network activity. And so what we're trying to do at System 1 is to harness and actually use that variability in order to drive a process towards neurotherapeutics discovery. And so this is our pipeline. We take blood samples from patients, and we get stem cells from those blood samples. Then we grow at scale in an automated system many hundreds of thousands or ultimately millions of organoids. We call this the farm. And the idea here is to be able to harness the variability and manage the variability of the organoids. So that requires that we measure across many different modes of neurobiology. So we use single-cell RNA-seq to measure the transcriptome. We use mass spec to look at the metabolome, the proteome, the lipidome, and so forth. We use many different imaging modalities to look at cellular cytoarchitecture, cellular morphology, tissue architecture, gross morphology. And then we're using the tools of electrophysiology and calcium imaging to characterize what are the neural network dynamics, what is the information that's being processed here, and how does that differ between disease and healthy. Ultimately, we get all this data, and we're left with this, we're suddenly in the data-rich regime, which is exactly where you want to be in the modern era, because once you're there, then you can apply and use and feel confident in the tools of machine learning. So you want to take all this data that you don't understand, put it into some configuration that you can understand, and say, okay, can we figure out what is the difference between our healthy and our diseased organoids? We call this process deep phenotyping. And what's the use of deep phenotyping for drug discovery? Well, once you have a deep phenotype, you can then start probing with either single compounds or combinations of compounds in this system and say, is there a way to make my diseased organoids look like the healthy ones according to the complex phenotypes that we've identified? This, of course, provides a path to clinical characterization. So this is where we're located in our facility in San Francisco. We've got a lab inside of a warehouse. We've raised $30 million in total, 25 last year. We have 35 scientists working on all these hard problems. And it is our core philosophy that we need to bring together organoid culture with automation and machine learning in order to unlock drug discovery in the neurosciences. Some of the programs we're working on include tuberous sclerosis, Rett syndrome, 22q11 deletion syndrome, and Alzheimer's disease. And we've got over 500 patient lines currently in-house. That's growing all the time. And we've got 100,000 organoids that we're currently characterizing. So let me just show you some results very quickly. So this is an organoid that we are able to evoke seizure activity in. So it's got ongoing spontaneous background activity right now. And in a few seconds, you're going to see large populations of neurons that are simultaneously active. Let's see, one's right there. See that? Another one. And then we add, there's the last one, then we add a known anticonvulsant drug, and you maintain the spontaneous background activity, but you get rid of the synchrony. Here's an example of where we characterize the neural activity in organoids that came from patients with schizophrenia and organoids that came from healthy controls. And we looked and we asked the question, how does the neural activity change in time in these two different organoid populations? And what we found was that the schizophrenic organoids, the neural activity elapsed much more slowly. It seemed to be perseverative in a way that we don't understand but might provide evidence into the underlying pathophysiology of this disease. The final thing I want to say is that we also study Rett syndrome. Rett is a very debilitating neurodevelopmental disorder that affects young girls. They lose motor function and language and have severe developmental delays for the rest of their lives. And we looked at the gene expression differences in all. We have one of the world's largest banks of Rett patient lines at our facility, and we look at the difference between genes in Rett patients and isogenic controls. And what we see is that there are definitely some that are upregulated, others that are downregulated. And if we compare them to what's known from post-mortem studies looking at tissue after death, in red across the top, those are all the genes that correlate between our study and those previous studies. But there are new genes here that haven't been identified that we're now exploring to see if we can get a greater handle on the underlying pathophysiology and hopefully mechanisms for treatment. And we can see very big separation between whether or not it's overexpressed or underexpressed in the healthy or control case. So this is the team, and I thank you very much for your time.
Did you get all that? So thanks, Sean. Sean's co-founder, Saul Cato, is also here. He's from UCSF, and some of the original science behind that project was created up there. And taking it back to the home team, another technology that came out of Stanford, Kunal Ghosh is the founder and CEO of Inscopix, an inventor of the miniature integrated microscope for in vivo brain imaging. This invention is already advancing fundamental knowledge of how the brain circuits function and underpin behavior, and is actually used by many of the scientists in this room today. So Kunal is a passionate advocate for the role of business as a force for good in society. He also holds degrees in electrical engineering from the University of Pennsylvania, a bachelor's degree from the Wharton School, a MS and PhD in electrical engineering at Stanford University. And so please welcome Kunal to the stage.
Thanks a lot for that introduction. It's great to be here. Inscopix, as was mentioned in the introduction, spun out from Stanford several years back. We came out of the labs of Abbas Gamal in electrical engineering and Mark Schnitzer in biology and applied physics. We're a company based on the miniature integrated microscope technology that was mentioned in the introduction that was developed to enable the means to read out large-scale neural circuit activity in freely behaving subjects. So following on the beautiful talk by Sean, our vision has always been to literally empower scientists to watch those neural circuits, those circuit phenotypes in vivo in freely behaving subjects across animal models. Inscopix today has commercialized the miniature integrated microscope. We have over 1,000 systems placed in over 400 sites across the world. Several scientists, academics in the Bay Area are valued customers and collaborators. We're developing a full stack sample prep through analytics to truly help the brain research community to mine the data and to understand the neural circuit basis of several brain functions and disease. Our customers and collaborators have collectively published over 100 papers in mostly top-tier journals, and many of these scientific publications I don't have the time to delve into, but really are starting to elucidate the neural mechanisms, the circuit underpinnings of basic brain function and dysfunction in the context of disease. We're just starting to gain some commercial traction. Super excited about continuing to empower the brain research community and continue to scale in this market, which is a difficult space. That's why we're all here today. Along the way, we were also extremely honored to partner with Tom Khalil and the previous administration as being an industry partner of the Brain Initiative. Technically, we're still a partner of the Brain Initiative. And also along the way, we're honored that WEF, the World Economic Forum, recognized Inscopix as a technology pioneer. So the miniature integrated microscope is nothing but a wearable on the cranium, on the brains of animal models, from mice to rats and now to non-human primates. It's the integration of a benchtop fluorescence microscope into a device that enables literally watching large numbers of neurons in real time. So here on the left panel, you're seeing the microscope mounted on a mouse model, and on the right panel, you're watching hundreds of neurons firing in real time while the animal is behaving. In this particular context, it's in the striatum, interested in understanding different motor behaviors and disorders. So similar applications with the platform are today deciphering the fundamental neural codes underlying memory formation, social behaviors, hunger, and a whole host of brain functions, with the eventual vision really here being that can Inscopix truly enable decoding of the brain? And then can we capitalize on the data and the platform to ultimately enable the development of precision neurotherapeutics in a very kind of organized and methodical way? I'm not going to repeat the statistics. I think several speakers today have told you about the challenges in developing therapeutics in this environment. It's the riskiest, costliest, longest. So our goal as we think ahead and we capitalize on the momentum we have and the data that we're seeing in several disease areas is, can we truly make preclinical drug development predictive of clinical efficacy? Mouse models, animal models in general, are notorious for being poor predictors of clinical efficacy. There has been a fundamental failure of translation from animal models to humans. We're making a bet that we can start to change the paradigm and we can move the needle by developing in-brain assays, circuit-based biomarkers that can transform this value chain, can at least increase probabilities of success. We want to bring pharma back in the game today. There is already an ecosystem of drug developers out there in big pharma, in smaller biotechs. They're fleeing neural, and the reason they're fleeing neural is because of this value chain being extremely disincentivizing for anyone to invest in. So we have a relatively modestly ambitious goal, which is, can we at least transform the economics here? Can we create more predictability in the process so that the existing drug developers and the drug developers of the future can get back into the game and can have predictability before they get into the clinic? As an exemplar demonstration of this, is just a proof point. There's a long way to go here. But in Parkinson's, together with Pfizer, when they still had a neuro program, another example of big pharma fleeing neuro. Inscopix, together with a lab at Stanford and Pfizer, demonstrated that the platform could, for the very first time, uncover what Parkinson's looks like in a mouse model of Parkinson's. So I don't have time to dig into the science here, but the Nature paper published last year shows literally a circuit phenotype of Parkinson's. So the next obvious question that the Pfizer team and drug developers had is, well, can this circuit phenotype be predictive of drug efficacy? So what we did, almost as an indirect demonstration that this had the potential of being predictive, is we took known good drugs and known bad drugs. So in the bottom left panel, what that cartoon is meant to show is that on all prevailing behavioral best assays, we have no means to predict drug efficacy. All those 3 drugs today, out of those 3 drugs, only L-DOPA is known to work. It's still the gold standard in Parkinson's. 50 years ago is when it was approved. The other 2 drugs have made their way into the clinic but are less efficacious than L-DOPA. All 3 drugs pass the behavioral test. They all correct behavior. The Inscopix data on the bottom right panel, again, difficult to appreciate from just the raw data, shows that only L-DOPA corrects the circuit phenotype and restores normal activity. So of course the question now becomes that can we start developing biomarkers not just for Parkinson's but across different disease verticals and start developing much more predictive models for drug efficacy than prevailing models. So we're taking, again, a very methodical approach. We are in this for the long haul. We are partnering with key disease researchers across different verticals, several of whom you already know, are friends, colleagues of yours in chronic pain, and continuing across Parkinson's and motor disorders, OCD, Alzheimer's, Huntington's, stroke, and of course the very difficult area of mental illness, looking at depression, schizophrenia. And the vision of Inscopix, just to close with, as we look ahead into the future, obviously we wish to continue scaling the research market, continuing to empower the research community. But at the same time, we want to start almost cataloging the data that is coming out of the research community. We're starting to think about how can we not only use the research community to help us continue to get key proof points across disease verticals, but can we truly build a cloud-based repository of circuit biomarkers that we can mine from these data sets? And then can we ultimately put it all together and start developing assays across these disease verticals which can lead to new screens for drugs as they are starting to approach preclinical to phase 1 and continued progression into clinical trials. So I'll close by saying that we all here are in the room today to hopefully start to usher in a new era in brain discovery and therapeutics. I think there's amazing momentum in the ecosystem. I've been doing this for more than a decade, since Stanford and then through Inscopix, and it's been really heartening for me to see the amount of activity in this space, both from the academic world and from the increasingly entrepreneurial communities that we're seeing being formed. We hope that Inscopix can be at the hub of this growing ecosystem, empowering neuroscientists, powering neuroscience discovery, and then hopefully we can start partnering across the value chain with developers of devices, and of course the existing drug development portfolio to leverage our platform and the data to truly start building new kinds of predictive models for drug development and for device development. Thanks a lot.
So now each of these companies is going to host a breakout table along with the other subject matter experts outside by the fountain in the same place where we had the speaker roundtables. That will be for the next hour. Then we'll head back in here for some closing remarks and some other surprises. So you're aware the NeuroLab is also open and available in the lobby area. And this evening, if you don't have plans yet, we've reserved 4 different restaurants for you to set up on conference dinners, and the sign-ups are on sheets outside by registration.